start recording. So it just remains for me to welcome everybody to the webinar this morning. And I do thank you.
To unmute your line, press pound 6.
A little bit of background here about the relationship between John Kulman and Bio-Rad. It actually started out about a year ago when John approached us at the PEGS conference in Boston on the East Coast here in the U.S. He was very impressed by the ProteOn and was very interested to see how our kind of 36 interaction array would work for him and how he could use it in his experiments to speed his time to results. And we were lucky enough to be able to place an instrument with John, and he's had the instrument now for 6 months, and he's been generating a lot of data for publications, and some of which he's presented at conferences, such as the Pep Talk conference earlier this year in January, and also the SPS conference, I think about a couple of weeks ago over in Phoenix. So we're very excited to have John as part of our 2010 Become an SPR Expert webinar series, and today he's going to talk about a method that he's developed, which is a calcium-dependent immunocapture strategy for Enhanced-Throughput SPR. And also John is going to touch upon thermodynamics and give us a little bit of a background as well to kind of, give us something to think about for the rest of the day. So with that, I'll pass over to you, John, and yeah, we look forward to hearing everything you have to say. Thanks, John.
Great. Thank you, Laura. So as Laura alluded to, I'm going to discuss a couple of things today. And the first is this idea of calcium-dependent immunocapture, which is a new way of doing SPR that we've developed here at the Puget Sound Blood Center. And then the later part of my talk, I'm going to get into how the ProteOn can be used to enhance the throughput. And this is particularly important because our experiments we're trying to do thermodynamic analyses, which involve doing SPR experiments at over a range of different temperatures, all of which takes time. So in order to get our data in a reasonable time frame, we wanted to enhance our throughput, which we were able to achieve with the ProteOn. So an overview of my talk today, I think this should break down into about 8 to 10 minutes per section here, but maybe a little bit less on the first one. But the first thing that I'm going to talk about is just some basics of SPR. Everyone that's here today and taking the time to listen to this is probably familiar with SPR. Secondly, I'm going to talk about how we developed calcium-dependent immunocapture and what that is and how it can be applied to a variety of different experimental formats. And then lastly, how we can apply this to do thermodynamic analysis on the ProteOn. So just the basic definitions, this is the only real basic slide here. When I talk about a ligand, I'm talking about the binding partner that is bound to the biosensor chip. And usually that, in conventional approaches, it's coupled covalently to the chip surface. And then the analyte is the binding partner that's in the mobile or fluid phase, which goes over the chip, and then we monitor its association with the bound ligand, all of which generates a series of curves, which we call sensograms. And when we're done with a single binding and dissociation series, then we have to regenerate the chip. And typically this involves post-run treatment of the chip with denaturants or pH extremes or chaotropic salts. And this is intended to remove any bound analyte but leave the ligand on the chip. And this is something that we have kind of figured out how to do differently. And I'll get into that when we talk about what calcium-dependent immunocapture is. So the rationale for what we want to do is that, in theory, SPR is an excellent technique for doing quantitative analysis of macromolecular interactions. But it's our belief that SPR is largely underutilized for a number of reasons, or I should say also improperly utilized. And some of these reasons are listed here. First cost and time limitations for producing a high-quality ligand. Usually that involves generating a ligand of high purity, a ligand of uniform conformation, and high physiologic relevance. And this can be quite involved. So first is generating the ligand. The second limitation is the difficulties that are inherent in regenerating a ligand-coupled surface between runs. And this is because if we use too harsh a conditions, that will destroy or diminish the binding capacity of the bound ligand. And if we don't use harsh enough conditions, we'll leave residual analyte on the surface of the chip, which will interfere with the subsequent run. And so, and the third limitation is the throughput limitation. And this is really where the use of the ProteOn comes in, where we use this 6x6 array to really enhance our throughput. And so the overarching theme here is that if we're able to eliminate these limitations, or at least mitigate these limiting factors, and take them out of our experimental design, then what we can do is do a very quantitative evaluation of macromolecular interactions very early in a development pipeline. So rather than wait until we have things that we're very interested in and generate ligands with high purity and do all of this work that takes quite a bit of time and energy, rather than do that later in the development pipeline, if we can eliminate the inefficiencies and the limiting factors and make this very quick and easy, we can do this on a larger number of targets earlier on in development. So in a typical SPR experiment, we begin by injecting some analyte onto the chip that has the covalently bound ligand on its surface. So the analyte is injected at a low concentration, and then buffer is injected over the chip, and that analyte is allowed to dissociate, and we monitor the dissociation in the sensogram shown here, and our units are here in resonance units. And when this is done, in this example, there's very little left at the end of the run, but this is the point at which one would typically scout conditions to do regeneration. And again, that involves detergents for pH extremes or chaotropic salts. And sometimes things like guanidine are used. So once this is done, then we go back and we do a second injection at higher concentration of analyte, and that's shown in the blue curve here. And we continue this process until we've generated a concentration series. And the sensograms that you see overlaid here constitute the complete raw data set. And in almost all cases, the most appropriate model for looking at interactions by SPR is the one-to-one or Langmuir binding model. And so what we do is we then use the software to fit the data to a theoretical model, which then will allow us, by doing this global fit to all the data simultaneously, will allow us to determine the equilibrium dissociation constant, or KD, for this interaction. So when we talk about doing SPR experiments, the SPR experiment itself is really kind of a minor component in the traditional way of doing this sort of work. So first, we have to start with a heterologous expression of a ligand to generate the recombinant ligand, and that can be done in insect cells, mammalian cells, bacterial cells, yeast cells. So there's a lot of different options here. If this is done in E. coli, for example, often, particularly with disulfide bonded proteins, this leads to insoluble aggregates that then need to be solubilized and refolded, characterized biophysically, to ensure that they are conformationally homogeneous and physiologically in a physiologically relevant conformation. And then we next need to go on to purify the ligands. So the expression and purification, and in some cases the solubilization and refolding, is where a large part of the investment of time and energy is prior to performing the SPR experiment. So once we have the ligand purified, we can covalently couple it to the chip. This is fairly straightforward. Typically, this is done by amine coupling, though there are ways of doing this. And then we do the binding experiment. So here, this is a sensorgram shown for a series of injections with regeneration at the end of each series, or at the end of each injection, and then this allows us to get the dissociation constant. And between each injection, we're doing regeneration. And when we finish this for this ligand, we then go back and repeat this process for a second ligand. And that second ligand may be a different ligand, or it may be a point mutation introduced into the original ligand, and where we want to see the effect of mutation on the interaction. So calcium-dependent immunocapture, turning to the next part of my talk, really is a strategy that we've developed in order to eliminate a lot of the difficulty and time of doing SPR. And essentially, here at the Puget Sound Blood Center, we're most interested in things that are involved in thrombosis and hemostasis. And those things are typically found in the blood, and they're extracellular, and they're human. And so we would like to make our ligands in the most physiologically relevant context, and that involves the use of mammalian cells, and we typically use CHO cells. What we do is we do we do transient transfection in CHO cells, and we append a short epitope tag to one terminus of the protein of interest, of the ligand, and do this in 12-well to 6-well plates. So this is a fairly small scale transient transfection of an epitope tagged construct. And the chip we then derivatize by conjugating a calcium-dependent antibody onto the surface of the chip. So here the antibody on the chip is covalently bound, and then it is used to capture in a calcium-dependent manner the ligand that we've generated by transient transfection. Now, when we do these experiments, rather than figure out ways to do regeneration here at the end of the cycle, since this is a calcium-dependent antibody, we just add EDTA, very mild conditions at neutral pH, and everything falls off. And then we can, on the same chip, put a new ligand down. So the difference between what we're doing here and the conventional approach is that in each binding and dissociation cycle, we're putting fresh ligand down each time. So we don't have any sort of degeneration of the ligand with subsequent cycles of regeneration. And the second thing to note here is that once we're done with this ligand, we then remove the ligand with EDTA, and we can put a completely different ligand or variant of that ligand down on the surface of this chip. So we can use this chip continuously for a variety of different ligands. So what is this calcium-dependent immunocapture? We begin with an antibody called HPC4. This is a mouse monoclonal antibody that recognizes human protein C, and the antibody itself binds calcium. And when the antibody is in its calcium-bound form, it can recognize a sequence of human protein C, called the protein C tag or the PC tag. And the sequence of that tag is 12 residues long, and it's shown here. So the first thing that we did was to do some fluorescence polarization experiments to just get an idea of what the equilibrium dissociation constant was that defined this interaction between protein C tag and HPC4. So we performed this experiment in the presence of 5 millimolar calcium at 25 degrees, and we were able to determine that the dissociation constant for this interaction is in the low nanomolar range. So I'd like to just take a moment to show a short video that demonstrates how calcium-dependent immunocapture works. Here we have the gold chip that is used in the SPR, and it's derivatized with carbohydrate. That can be carboxymethyldextran or alginate that's been derivatized with carboxylate groups. And underneath here is the optical unit of the SPR instrument, which is measuring the amount of mass that accumulates on the surface, and there's deflection of this beam here as mass accumulates on the surface. So as this goes on, you may notice that this beam is moving back and forth across the detector surface. So the first step is to couple the antibody to the surface of the chip. So here we're doing just a standard amine coupling of the antibody to the chip, and then we're adding calcium. And calcium causes the antibody to change conformation such that it can now bind to that epitope tag. Now what we're doing is injecting the crude serum-containing conditioned medium from cells that are making the ligand with the protein C tag on it. And so the ligand of interest is shown here in green, and the protein C tag, that 12 residue peptide, is shown here in blue. And in the presence of calcium, the antibody will grab this from solution. And effectively what we're doing is on-chip purification. So we eliminated purification from the experimental design because it happens as a step in the SPR experiment. So we capture the ligand, and when the ligand is now bound to the surface of the chip, we now inject the analyte, in this case an FAD fragment. And the analyte binds. The sensogram shows a deflection as this binding occurs, and then we allow buffer to flow over the chip and for the analyte to dissociate. And you'll notice that this deflection goes back down. Now when we're done with this, we add EDTA, which releases calcium from the antibody, which releases the ligand and analyte-ligand complexes from the surface, and we simply add back calcium and can repeat the cycle over and over and over again. So what I've shown so far is basically a general mechanism, but specifically what we're interested in is a molecule called von Willebrand Factor, and we're interested in evaluating monoclonal antibodies that bind to a part of von Willebrand Factor which have antithrombotic potential. So we are, after all, a blood center here, so we're interested in how we can prevent an occlusive thrombus from forming, which would lead to heart attack, stroke, pulmonary embolism, and those sorts of things. So I'd just like to take a few moments to talk about the ligand. It's the von Willebrand Factor A1 domain, and this is the von Willebrand Factor here. It's a large multi-domain glycoprotein in the blood, and the A1 domain is shown here. Von Willebrand Factor, what it does is it is a bridge between the damaged blood vessel and platelets. So it grabs platelets and causes them to aggregate at the site of vascular injury. And how this works is that when a vessel is ruptured or a plaque is ruptured or there's otherwise some damage to the vessel endothelium, this exposes collagen that's present in the subendothelium. And this collagen is recognized by the A3 domain of von Willebrand Factor. Next, the A1 domain binds to a protein called glycoprotein 1b alpha. And this is on the surface of circulating platelets. And this is the way that von Willebrand Factor serves as a bridge between platelets, which are in circulation, and the damaged blood vessel. And this results in the formation of a thrombus. Physiologically, this is what happens, and pathophysiologically, this is what happens to lead to an occlusive thrombus forming. And this is the process that we want to evaluate modes of intervening to prevent this thrombus from forming. So the analyte that we're going to use, so A1 domain here is the ligand. And the analyte is an antibody called NMC4, which binds to the A1 domain, and it prevents the binding of the A1 domain to glycoprotein 1b alpha on the platelet surface. And the result of this is that the thrombus does not form. So to give you a little more microscopic view of what's going on here, if we take a look, this is the crystal structure of the isolated A1 domain of von Willebrand Factor, shown here in yellow. And shown here in red is a fragment, the relevant fragment of the platelet glycoprotein 1b alpha. So this is the receptor on platelets in red, GP1b alpha, and it's binding to the A1 domain, which is part of this is the tether that links platelets to the damaged blood vessel. And the antibody that we're interested in is called NMC4, and it's shown here in green and in blue. And in blue is the heavy chain of the antibody, and in green is the light chain of the antibody. So what we want to do is look at this interaction to determine the equilibrium dissociation constants, and then do more sophisticated mutagenesis studies and evaluate the thermodynamics of this interaction and see what is the effect, what is the contribution of each of the residues within this structural epitope. And so to define, first of all, what is the functional epitope, and then more carefully define the contribution of each residue. So that's what we'll be talking about here today, is evaluation of what's going on in this interface. Okay. So the first step here then is the functional epitope mapping, and shown here on the top is the sensograms for the wild type A1 domain here binding to the FAB, the antibody fragment here. And this may take just a moment to load. So if we go back, shown here in surface representation is the heavy chain of the antibody in blue, the light chain of the antibody in green, and then in the gray surface here is the A1 domain. And if we take a look at a ribbon diagram here, we can see a little more closely what's going on. The first thing that we did was to mutate every residue in the A1 domain that's within a 3.5 angstrom radius from the surface and making contact with the FAB fragment. So there's 4 residues here on this one side that fulfill those criteria. And then if we turn the molecule around, we can see that there's another 4 residues. So the first thing that we wanted to do was to mutate each of these residues to alanine and see the effect of substitution on the binding. And then we introduced a number of other point mutations that were somewhat distal to the binding site, close but not part of what appears to be the structural epitope. And then we introduced a number of other mutations at the other end of the molecule. So these and these should serve as negative controls. And so this represents the 18 different mutations that we generated for these studies. Okay. So the first thing, looking at the controls, if we look at the residues that we mutated that are distal to this binding site, we see that the dissociation constant is of the same magnitude as the wild type. So this is what we would expect because these are not within that interface. If we then take a look in the interface, there's 2 residues here which appear to be making contacts in the crystal structure, but when mutated to alanine, do not seem to affect the binding of the A1 domain to this FAB. So this is an example of things that are in the structural epitope but are not part of the functional epitope. Next, we mutated these 2 residues, both of which are arginine residues, and we saw that mutation of either of these residues to alanine has a profound effect on the binding, completely abolishes binding. Now, if we take a look at the crystal structure, some of this would make sense, and some of it wouldn't make sense if we were just looking at the crystal structure alone. If we look at this first arginine residue, and we zoom in here, we see that it's making modest contacts with the heavy chain. And one might predict from this that mutation of this residue to an alanine would have a modest effect on the binding, when in fact, when we do the SPR experiment, we see that it completely abolishes binding. If we now look at the other arginine residue, which is over here, we see that it's actually making a variety of different contacts with both the heavy chain and light chain residues. And so this one makes a little bit more sense that mutation of this would abolish binding. So moving on to the last 4 residues which are in this interface, these 4 residues, when mutated individually to alanine, affect, one might notice that they affect not so much the association rate, but rather increase the dissociation rate. And this is the basis for their lower affinity We're seeing things in the 6 to 8 nanomolar range relative to 1.7 nanomolar for the wild type. So here we've been able to use calcium-dependent immunocapture to analyze the effect of mutation on binding of these variants. But what we would like to do is to do thermodynamic analyses and more carefully define the contribution of each individual residue. And we want to define that in terms of the main thermodynamic parameters, which are free energy, enthalpy, and entropy. And so I'd just like to take a couple of moments to explain what thermodynamics is and how it relates to SPR. Basically, thermodynamics, a central tenet of thermodynamics is that free energy for a spontaneous process is negative. And we probably all remember from general chemistry that delta G, the change in free energy, equals minus RT ln K. And in this case, the K that we're talking about is the equilibrium dissociation constant. And R is just the gas constant. And K is the absolute temperature in Kelvin. So this is an important relationship here. It's the delta G equals minus RT ln K. But in SPR, what we observe are the rates of association and the rates of dissociation. And so we want to be able to get from the rates of association and dissociation to determining the equilibrium dissociation constant, and from that to get to delta G. So for a simple reaction, such as the one that we're looking at here between an FAB fragment and its ligand, A plus B forms a complex called AB, and this is an equilibrium. And by definition, the equilibrium dissociation constant is the concentration of A times concentration of B all over the concentration of the AB complex. Now, also from general chemistry, the rate for the forward reaction, or the rate of association, is defined by the forward rate constant times concentration of A times concentration of B. And for the reverse, the rate is equal to the dissociation rate constant times the concentration of the complex. And importantly, at equilibrium, the forward rate and the reverse rate, by definition, are the same. So nothing is changing because it's at equilibrium. And so we have this relationship, is that the forward rate equals the reverse rate, and therefore this equals this. And from this, we can do just some simple math and come up with an important relationship, which is that the equilibrium dissociation constant is equal to the dissociation rate constant over the association rate constant. Okay. So there's only a few more slides here on all this, so please bear with me. So why is this important? Well, SPR allows us to determine this and determine this, and from this we can derive this. And if we can derive this, we can then determine by the relationship delta G equals minus RT ln K, we can determine the change in free energy. But the free energy is also defined in terms of the contribution of enthalpy and entropy down here. And so what this allows us to do is a type of analysis called Hoff Analysis. And in a nutshell, we can express the dissociation constant in terms of enthalpy and entropy. And what this really means for practical purposes is that we perform SPR experiments over different temperatures, over a temperature range. And if we plot the natural log of our dissociation constant from each temperature as a function of one of over the temperature, the absolute temperature, we generate a line, and the intercept of this line tells us the change in entropy, and the slope of this line tells us the change in enthalpy. Similarly, so that would tell us something about this change here between the unbound and the bound form. But if we want to learn something about how it gets from here to here, That is, how does it get from the unbound state to the transition state to the bound state? We can apply statistical mechanics and determine this double dagger here represents the transition state. So we can determine energy differences here, and we can determine energy differences here. Simply by performing our SPR experiments over a range of temperatures and then plotting them appropriately as a function of 1 over the inverse temperature. Okay, so the big picture, to kind of put all that business behind us, the big picture is that we want to determine the changes in free energy, enthalpy, and entropy for an interaction. In this case, an interaction between an antibody and a ligand. And then we want to perform the same experiments after making amino acid substitutions, and then see what the effect of those substitutions is on enthalpy and entropy and free energy. And if we can determine these relationships, we can then use this information to develop protein-based therapies with desired characteristics, perhaps to develop hypotheses to enhance association, or more importantly, or more relevantly, to slow down dissociation of, say, a therapeutic monoclonal antibody. So, as you can probably guess from the last few slides, it's very important to perform these experiments at different temperatures. And this involves a certain amount of time to do these experiments at each temperature. And so these experiments could get extremely long. So this is where we turned to the ProteOn to use the capacity of the instrument to do multiplexed interactions simultaneously to really cut down the throughput time. And so for those of you who aren't familiar, this is the how the ProteOn is set up, where we have a chip, but then there is a fluidics channel. There are fluidics channels that run over the surface of the chip in one orientation, and we can then use these to capture 6 different ligands on the surface of the chip. And then by rotating the fluidics, we can then inject in perpendicular orientation a concentration series. And what this allows us to do is simultaneously monitor the interaction of 36 different things on one chip. And the data set that would come out of a single run would look something like this, okay, where we've included here a reference channel. So there's potentially 36 different interactions that can be looked at. In this case, we're looking at 30 different interactions. So some actual data. Here what we've done is to put the von Willebrand Factor A1 domain, our ligand, capture it on the surface of the chip. Here we're only looking at the wild type A1 domain. And we tested the effect of different immobilization levels. So here we've gone from a low level to a fairly high level of immobilization of the ligand, and then we've done a concentration series. So this here represents one single run on the ProteOn, where we've generated these 5 different panels, and each one represents a different amount of immobilized ligand. And then the first thing that we wanted to do was to get an idea of the reproducibility run to run. So we've repeated this experiment 6 different times, one right after the other. And what you can see is that for all cases, we see a very reproducible data set. And you may notice that there's a red line. The red line is the application of the theoretical model for one-to-one binding. And so what we can do is then measure this chi-squared value, which is a measure of the fit of the data to the line. So the other thing that we were able to do is, since we did this in 6 different replicates, we were able to derive meaningful statistics, which would tell us something about the error in our measurements. And if we look here, as we go from high to low amount of captured ligand, we see that at this point, our standard error is the lowest. And we have a fairly reasonable chi-squared value, which is a measure of the fit to the data. So this was the immobilization level that we used in all subsequent experiments. So next, we wanted to see what would happen if we perform these experiments at different temperatures. So here we've taken the wild-type A1 domain and put it in all 5 available channels, and then we've done an experiment and then moved in a 2.5 degree increment from a range of 15 degrees Celsius up to 35 degrees. And we see that so this we have, in this experiment we have parallel replicates, so these are all done at the same time. And we see that there's a nice agreement when you go top to bottom in one column of the data in parallel replicates. So lastly, what we wanted to then do is to see the effect of mutation and then derive our thermodynamic parameters. So at 15 degrees, we did a single experiment. Here we put the wild-type A1 down, and then we put these 4 different mutants on the same chip, but in different channels. And we performed this experiment at 15 degrees Celsius. And this entire experiment took about 30 minutes. So having done this, we wanted to, again, generate meaningful statistics. So we replicated this experiment serially 5 different times. And this entire series at 15 degrees took 2.5 hours. And then we simply changed the temperature of the instrument from 15 degrees to 17.5 degrees and generated the same sort of data by performing this serially 5 different times. And then we continued at 2.5 degree increments all the way up to 35 degrees. So here we've generated kind of a multiplexed array of our different mutants in pentuplicate over a range from 15 to 30 degrees with 2.5 degree increments. And this entire experiment took about 32 hours of runtime. And this corresponds to upwards of a month of machine time if one were to be using conventional approaches. And what this allows us then to do is to derive actual data, the Van Hoff analysis, to look at the differences in enthalpy and entropy upon binding, but also the transition state analyses that I talked about, or the Eyring analyses, which allow us to determine the energetics of the transition state. And we did this for all 5 different constructs, the wild type and the 4 mutants. And once this is done, we can express these data in a tabular form. And here, I apologize, but this little box should be a double dagger to represent the transition state. So we can determine the free energy for equilibrium for the transition state going forward and reverse. We can do the same thing for enthalpy. We can do the same thing for entropy. And we're doing this all for a variety of different ligand variants, the wild type and the 4 mutants. And these can be expressed graphically here in terms of free energy, enthalpy, and entropy to look at the binding coordinates. So here we have the unbound state, the transition state, and the bound state in terms of free energy, and the same sorts of things in terms of the enthalpy and the entropy. So to summarize, there's really two main parts to what I've talked about today. One is the calcium-dependent immunocapture. To reiterate, we're able to generate our ligands by small-scale transient transfection, which means that the rate-limiting step in generating our ligand is actually just making a plasmid. This is transiently transfected into CHO cells, which takes about 5 minutes of hands-on time. And after 24 hours, the medium is harvested and injected onto the chip. There's no upstream purification of the ligand necessary. And the chip can be regenerated very mildly and simply with millimolar concentrations of EDTA. And we can use the same chip for multiple different ligands in a continuous mode. The second part of my talk here has been the use of the ProteOn to enhance the throughput to enable highly reproducible thermodynamics experiments to be done in both parallel and serial replicates in a reasonable amount of time. And this compares very favorably to conventional approaches because we take advantage of the fact that we're doing multiplex analysis, looking at 36 different interactions at a time rather than a few. And I've shown that the data are reproducible in parallel on the same chip in different channels, but also serially if we do them one after another subsequently. We see that the experiments are highly reproducible, and we can generate high-quality thermodynamic data with fairly low standard errors. So with that, I'd like to wrap things up. I'd like to acknowledge the contributions of the people, first and foremost, in my group here at the Puget Sound Blood Center, Jeff Harris and Jasper Lynn, who work in my lab, and Doug Bolgiano, who is a statistician who helped with the analysis of this. My colleague Greg Mize at the Fred Hutchinson Cancer Research Center here in Seattle, our colleagues in Japan who provided the NMC4 monoclonal antibody, and funding from the Puget Sound Blood Center and the National Institutes of Health, and lastly, the support of the good folks at Bio-Rad, Laura Moriarty and Jill Raymond, and Mohammed Youssef, who've been extremely helpful in getting us up to speed and helping us to place an instrument here to really test out how we could get our work done in a much quicker way than we had been doing. And I guess I wouldn't want to end this without acknowledging the work of the people who did the protein structures that I've just kind of thrown up in these PyMol animations, but this really represents a tremendous amount of work over a number of years. So there's all these publications that deserve mention here from different groups in the U.S. and in Europe and in the UK. And with that, I guess I'll go to the last slide here, and I don't know if I think I'll turn things back over to Laura at this point.
Okay, great. Wonderful. Thanks ever so much, John. That was brilliant. I really enjoyed that. Got my brain going in terms of the kinetics and the thermodynamics again, and I hope it did for everybody else. So what I'm going to do right now is I'm going to go ahead and unmute everybody so that people can start asking me questions, because I'm sure there is hundreds of questions. So everyone, you are unmuted. So please go ahead. If you have a question, please go ahead. No? Okay, well, I'll start everyone off.
Okay.
So you've used this method with this kind of calcium-dependent switching mechanisms to change the structure of the antibody and then, remove the binding effects. Is that something that you think could be applied to, is that like a big class of proteins that have that kind of switching mechanism for structure in terms of
well, I think
calcium binding?
The antibody is probably more of a rarity than anything else. But the antibody itself would be the common thread if one were to be analyzing different ligands. So what I'm getting at is that, well, I think that calcium-dependent binding is a fairly common biological theme. In antibodies, I don't think that it is, but by using this antibody, simply coupling this particular antibody, which is commercially available, to the surface of the chip, we can then append the same epitope tag to any ligand that we want. So really the calcium-dependent immunocapture is not specific to our interaction that we're looking at here. It's specific to the interaction between that antibody and its tag. What one chooses to put that tag on is up to them. So I think it's fairly widely applicable in SPR and in other capture strategies. It can be used for just about any conceivable ligand-analyte interaction.
Oh, that's great.
That answer your question?
Yeah. So that's great. So you're really kind of opening up a new kind of corner of the world there for folks who are doing SPR. That's great.
Yeah,
well,
we're currently trying to apply it to some more challenging ligands, such as, there's nanolipoprotein particles that we want to be able to capture transmembrane proteins, and specifically transmembrane proteins which are in a physiological context and conformation. So if one were to be looking at antibodies directed against G protein-coupled receptors, for example, as a therapeutic mode, the trick is to get the GPCRs in a lipid context where they're in the right conformation, and then get them on the chip. And so we're now kind of pushing to see if we can use this calcium-dependent immunocapture strategy to capture more what have been more recalcitrant type systems. To see if we can get it to work for those.
Oh, it's funny you bring that up because we were working with a company called Integral Molecular that have the lipid particles. So maybe we can talk a little bit. I'll send you some information about those. They might be something that might be useful for your GPCRs.
Yeah,
there you go. We learn something, you learn something. Okay, so anyone else out there have a question?
Yeah, I have a question.
Oh, great. Go ahead, Tamar.
This is Dalia.
Oh, it's Dalia, sorry. Whoops.
No problem. Similar accent, I think.
Yes.
Yeah. Okay, so John, this is regarding the mutants you did. What did you do them deciding on the residues only based on the structure, or did you use also the sequence and whether conserve the residues?
I see. So, we looked at conservation in other vertebrates of residues, but to see what seems to be important in this interaction between the A1 domain and glycoprotein 1B alpha. But the main criterion for mutagenizing residues was to simply put it into, we just went into Swiss PDB viewer and selected every residue on the ligand that was within 3.5 angstroms. So it's possible that there are residues that are not in that interface that are more than 3.5 angstroms away, which are involved in some sort of conformational stability that would impair the binding. But we just did a quick grab of all residues which were within 3.5 angstroms, and that was just a distance that we chose, which is fairly standard. But I chose that distance based on the crystal structure. Yes.
Okay. And the results did they show that these residues were indeed involved in this interaction? How did they affect it?
So to summarize, we saw that there were 8 different residues which are in that interface on the side of the ligand. 2 of those residues, which were in 3.5 angstroms, did not have any effect on binding. 2 of those residues completely abolished binding when they were mutated. Those were the 2 arginine residues. And then 4 of the residues, when mutated, led to diminished binding. So if we just looked at the crystal structure, we would say, okay, 8 residues are involved. This is the structural epitope. And really what this enabled us to do was to say, of those 8 residues, 2 are important, 2 are absolutely essential, 2 are not important at all, and 4 are of intermediate values. So, and that's something that we wouldn't have been able to discern just on the crystal structure, the structural data alone. So,
okay.
Does that answer your question?
There are some, yes, very much. Are you familiar? There are some servers that combine the structure together with the sequence similarity or conservedness of the residues on the interface between two proteins. So they combined it, and then you can see together which residues should most influence the binding. Did you know them? Did you use them?
We haven't, no, and I think we're familiar. My postdoc is a structural biologist, so he does a lot of this stuff. But but I think what we determined was decided was we're not looking at the interaction between this two proteins for different organisms, for different species. What we're looking at in this case is an antibody specifically binding to this A1 domain. So that sort of approach, I think, is more of interest to us when we are looking at another project that we have going on, which is actually looking at the interaction, the thermodynamics of the interaction between the A1 domain and the platelet receptor. So there, we can look at that interaction in different species. You know, we look at the sequences of both of those binding pairs and then look at it in zebrafish, we can look at it in fugu, we can look at it in dog and so forth, and really determine, you know, where we would fine-tune our mutagenesis and then kind of go from there. So, yeah, I think that's an excellent approach. For this one, we're looking at a monoclonal, which is sort of an artificially generated thing to begin with. It's not the product of eons of evolution. So, so we didn't look at it that way. But it's an excellent point, and I think one that we're interested in for looking at other interactions. Yes.
Okay, thank you. It's a really nice work and very interesting.
Yeah, thank you very much.
Great, thanks, Dahlia.
Hey, Laura.
Yeah.
Hey, Laura. This is Ruben. Hey.
Hey, Ruben.
Hey, John. Okay. Hi.
Hi,
John. Yeah, thank you very much for showing the great work on thermodynamic analysis.
My
it's the first time to run that. Yeah, it's great. I just have a quick technical question. So when you do a natural log on the coefficient, the equilibrium coefficient, or rate constant, do you need to make the value first dimensionless? I mean, just to remove the physical unit.
No. I mean, the units are irrespective of whether you've taken the natural log. Yeah. So as long as, well, I don't know if I can go back to the slides. I think I can. Can you see my presentation right now?
Yes.
Okay, I'm still on then. Okay. Yeah. So I think what you're getting at is
I think, you have one slide showing there's, I mean, maybe just 2 or 3 slides later.
Of actual data?
Yeah.
Okay. I think maybe you're talking about this one
here.
There?
Yeah.
Yeah.
You have a unit there, log Ka over T.
Right. And so the units here, have in units of moles and then mole seconds and temperature. So yeah, it gets kind of complicated, but it does add. I mean, if we look, for example, here at the ln of the K d, the dissociation constant, and then see what's the best. I'm trying to think
I see.
Where I would find this.
Okay, I kind of understand. So you apply, I mean, the same, in all analysis, you apply the same unit in the scale, so kind of like normalized to a standard. Like standard. So, yeah.
Exactly, because what we end up with is that the slope is in units of energy, and the intercept is in units of energy. So what we want to have is, we basically want to have this to be in constant units, which gives us kilojoules per mole. So they're all sort of, I don't know if you'd say normalized, but they're all in the same unit ultimately when we get done with this. But
I see. Yeah, I got it. Okay, thank you very much, John.
Sure, no problem.
Anyone else out there have some questions?
Hey, John. This is Hugo. I have a quick question. Hi, John.
Yes.
This is a question regarding the, you have 2 arginine, when you mutate to alanine and abolish the bonding. So do you think it's possible those 2 arginine, when you change to alanine, not only may change the structure a little bit, but maybe even move the rest of the residue off the position so that abolish the bonding? maybe?
That's a good question. I think that we, particularly with arginines that, have a capacity to form salt bridges, there is a question of whether they're involved in sort of the conformational stability of the molecule in unbound form. But if we take a look, one of the things that we notice is if we can go in here and imagine that we don't have the antibody. Are you able to see my presentation right now?
Yes, yeah.
Okay, so the heavy chain here is in blue, and then the light chain here is in green, and this is one of those first arginine residues. And if we take a look here, we'll see that this is largely just sticking out into solvent, and this is in it's not making any. If we can imagine that these things aren't here, it's not making any contacts with, say, acidic residues in the ligand here. And we also, I mentioned in the acknowledgments the 4 different structures. This is also the case in the crystal structure of the free ligand. Okay, so without this molecule here, we can look at this, and it's basically an arginine that's sticking out directly into solution into the solvent. And if we then take a look also at the second arginine, it's also very much one that you can kind of see here is projecting straight into solution from this helix, and it's not making any contacts with other residues in the molecule. So that's an excellent point, and one that that we were concerned about, that the mutation could destabilize the molecule. But we're currently working on some other monoclonals against this domain to really confirm what we're assuming here, and that is that if this, for example, mutated to alanine causes destabilization, it should affect the binding of an antibody that binds elsewhere but in proximity to the molecule. So that's something that, again, we want to be able to do in a multiplex format if we're looking at multiple antibodies binding to particular mutants, that we could do in the proteon by, we could conceivably look at 5 or 6 different antibodies binding simultaneously on one run. But it's an excellent point, and I think it's one to consider seriously, that these mutations could destabilize the domain and give you those results. But we don't believe that that's the case here.
Okay, thank you.
Does that answer your question okay?
Yeah.
Okay, good.
I think there was another question there.
Yes.
Yes, this is Perry.
Hey, Perry.
Yeah, wanted to see if you had selected the calcium-dependent antibody capture method because there were some others that just were not as fruitful or advantageous, and do you think that this system has some advantages over some of the other types of antibody capture methods that are being used today?
Well, I think we've actually not directly used other antibody capture methods because this did work pretty well from the very beginning. This is the way we went. But even if one is looking at an antibody that's, there's a number of different approaches that are used out there where one uses a capture antibody. If in our situation, we're putting EDTA on to regenerate the antibody and release anything that's bound to it, and that we've shown we can do for hundreds of cycles without loss of binding ability. Now, if we did the same thing even with an antibody, which tends to be fairly rugged, but we were exposing it to magnesium chloride or SDS or pH extremes, there's going to ultimately be a degradation with time, and that's going to be something that maybe you wouldn't be able to get 100 different cycles out of that chip. So I can't speak to that from direct experience, but my colleagues have had pretty good success with other antibody capture strategies. But I would add that they're not doing the kind of throughput that we're doing. We really want to be able to do something ultimately where we can run the proteon for days to do a whole bunch of different stuff, and we don't want to have to make a new chip and put a new chip in the instrument. We don't want to have any concerns about chip-to-chip variability. We want to run all of our experiments on one chip. So I prefer this method. We have tried some strategies where we've biotinylated ligands and put them onto streptavidin derivatized chips. And that works very well, but again, you're still running into the limitation of regeneration. So to my knowledge, this is the only technique that's shown to be robust in terms of capturing the antibody and quantitatively capturing the ligand and retaining it throughout the course of the experiment over a wide temperature range. We don't see it coming off when we go up to 35 degrees. It stays on. And then it's quantitatively released when we put EDTA on. So I hope that answers your question. But
no,
I don't have direct experience, but my postdoc has done a lot of that sort of work, and there's a limited longevity when you do other immunocapture strategies where you have to do a more standard regeneration.
Right. Yes, I would have guessed it would have been more gentle way to go compared to some of the other strategies. I just thought you might have had some quantitative data that would be beneficial to others. But yeah, that several hundred or 100 regenerations is pretty good for those types of capture methods.
Yeah, we're kind of surprised actually at how well it worked.
Cool. Great. Thanks, Perry. That's a great question. So I'm going to ask if there's just one more question out there. Nope. Okay, great. Well, just to remind. Oh, that's it.
Okay.
All right.
Well,
so
I can go get my breakfast now.
You can have breakfast and coffee. And thanks everybody very much for your kind attention this morning. And I hope everybody learned something. We're going to be posting this recording on our box.net server, so people can download it at their leisure. And yeah, thanks everybody, and have a great day. Thank you, John.
All right. Thank you.
Bye-bye.
Bye-bye.